All right, everyone. I think I've got this recording, so other people can access this later, or you can access this later if you'd like. All right, so do I have any burning questions? Anybody want to just start off with some questions, or do you want me to just systematically go through a lot of the lectures like I did last time? Just go through the lectures. Okay, and then, yes, I got your email, so I will post those top hat questions right afterwards. Yes. Okay, so, all right, well, let's just start with lecture 7. Oh, you have, okay, yeah. Sure. Can you just read it off for me? Oh, okay, sure. So the practice exam is a 25-year-old man complains that his muscles are stiff after he exercises. When you ask him to flex and extend his arms rapidly and repeatedly, you notice that he soon slows down and his muscles seem to remain tense and contracted longer and longer. So he's able to extend and flex his arm initially, but then after a while, he becomes stiff and he can't relax. The impaired skeletal muscle relaxation could be explained by a defect causing reduced activity of which of the following? Either A, the Circa pump, B, dihydropyridine receptors, C, voltage-gated sodium channels, and D, ryanodine receptors. So just by looking at those, which one is not like the others? 3 of them would, with reduced activity, would cause muscle weakness, not stiffening. Only one of them is going to cause a stiffening in the flex and extension response. What do you guys think? Circa pump, dihydropyridine receptors, ryanodine receptors, or voltage-gated sodium channel? Circa pump. That's exactly right. Okay, so the idea here is the Circa pump is responsible for resequestering calcium. And without the Circa pump, you're not going to get relaxation because calcium will stick around in the intracellular space longer. Okay? So reduced activity of the voltage-gated sodium channel is actually going to cause muscle weakness. Same with the dihydropyridine receptor or the ryanodine receptor, because those are responsible for initiating that calcium influx into the intracellular space. Only the Circa pump is responsible for removing the calcium from the intracellular space. Okay, does that make sense to everybody? Do you want to just go through that whole event? That's kind of lecture 7. So let's go ahead and just go through the entire, and what I may do, I know this is really kind of annoying for the people at home, but what I may do is write a lot of this on the board, okay? So with lecture 7, just remember there are some things in common that all muscle cells have. They all are considered a sliding filament mechanism because all of them, smooth muscle, cardiac muscle, and skeletal muscle, involve myosin and actin, okay? So the myosin is the molecular motor. It uses ATP to ratchet along the actin, moving, causing contraction. Okay, so it's the basis for shortening. Also, these all 3 types, they have this in common. Calcium is the key. Calcium is what initiates the contraction in all 3 muscle types, skeletal, cardiac, and smooth muscle. Okay? And then they all have an electrical component. Okay, so they're linked to, this is called excitation-contraction coupling. Okay, So a lot of you guys have had this in laboratory, but I do want you to know some of the morphology. Okay, so I do want you to know some of the major differences between skeletal, cardiac, and smooth muscle. Remember, skeletal and cardiac are both striated. Okay, they are organized in sarcomeres. Smooth muscle isn't. Okay, so this is just general information about muscle. Striated, those are the skeletal and cardiac muscle. Smooth muscle don't have striations. All right. Also, two main types of control: the voluntary system, that's the somatic nervous system, that's a skeletal muscle, right? You have control over your skeletal muscle. Involuntary, that includes the autonomic nervous system, the sympathetic or parasympathetic nervous system. Okay, so hopefully you guys also were able to find that whole list of definitions. Hopefully you were able to go through a lot of them. I'm kind of going through a lot of them right now. One of them is a motor unit. What is a motor unit? Remember, a motor unit is a motor neuron and all of the muscle fibers that it innervates. Okay? Single motor unit consists of a motor neuron and all of the muscle fibers it controls. All right, knowing a little something about the anatomy of a muscle is important as well. So we already went through some of the terms. These are also on that list of definitions. You should know what a sarcolemma is. Okay, sarcolemma is the plasma membrane of the muscle. Okay. What is the sarcoplasmic reticulum? That's essentially the endoplasmic reticulum. But when it's in muscle, you can call it the sarcoplasmic reticulum. T-tubules are just invaginations. They're folds of the sarcolemma. It dives deep into the interior of the muscle. Those are called the T-tubules or the transverse tubules. And the terminal cisternae of the sarcoplasmic reticulum, it's part of that compartment, the sarcoplasmic reticulum, but it's the part of the compartment that is right up next to the T-tubule. Okay, so that's called the terminal cisternae. Okay, so just a few definitions. If anybody has access to that definition sheet and you notice that I'm not covering some of the definitions, just let me know and I'll make sure I cover it. So if you need some text associated with these terms, this kind of gives you some information again about the T-tubules and the sarcoplasmic reticulum. Remember, all of the calcium is housed inside that sarcoplasmic reticulum. And when that muscle cell is excited, remember these are excitable cells, the calcium is released. Ultimately, it's released into the intracellular space. Okay? And that intracellular space is also called the sarcoplasm, which is kind of listed right here too. So another review slide, but it shows you the sarcolemma is the plasma membrane. Sarcoplasm is the cytoplasm. That's the intracellular fluid of the cell. And the sarcoplasmic reticulum is really the smooth endoplasmic reticulum that houses all the calcium. Okay, so this is where we start. This is like, again, the chain of events that occurs from the motor neuron all the way to contraction of that muscle. Okay, so it starts off with the action potential. This is from exam 1, propagating down the axon of the motor neuron. It arrives at the axon terminal end. So I think you guys can see my arrow there. This whole part of the neuron is called the axon terminal end. And that change in voltage, right, because it's the action potentials that are propagating down the axon, it arrives at the axon terminal end, and that voltage depolarization, that voltage change, actually opens up voltage-gated calcium channels, right? So calcium floods into that axon terminal end. Calcium binds with all of these, well, it's a docking protein that causes vesicle fusion and the release of acetylcholine into the synaptic cleft. So acetylcholine is the ligand, it's the neurotransmitter that diffuses across this synaptic cleft here. And let me know if you're not understanding some of these terms. I know these are sometimes this is really review for a lot of people, but sometimes this is brand new. And so I want to make sure that you guys are understanding the terms. Acetylcholine is the neurotransmitter that's released into this area here called the synaptic cleft. And the neurotransmitter acts as a ligand binding to these nicotinic acetylcholine receptors. Now there was a quiz question, I don't know if you guys remember that, and one of the foils was muscarinic acetylcholine receptors. Remember, those are in the heart. So with skeletal muscle, these receptors are called nicotinic acetylcholine receptors. And they are ion channels. Unlike the muscarinic acetylcholine receptors in the heart, which are metabotropic, they're G protein coupled receptors. Okay, so acetylcholine binds to the nicotinic acetylcholine receptors and opens up a non-selective cation channel. It allows for both sodium and potassium to move across the membrane, which essentially depolarizes the cell, right? So hopefully this is review for you guys. You're just kind of, I'm just walking you through it. So it depolarizes the cell because now the relative conductances for sodium and potassium are 50-50. The membrane potential moves towards zero, right? Depolarizing this area called the end plate. So these end plate potentials are basically graded potentials. Everything you know about graded potentials applies here. They decay with distance, right? And they don't need, there's no summation here. It's enough of a depolarization that ultimately it activates voltage-gated sodium channels right outside the end plate, which initiates action potentials. Okay, so action potentials are then initiated right here outside the end plate. And that's due to, again, the activation of voltage-gated sodium channels. Any questions, you guys, so far? Okay, so that's kind of what happens at the neuromuscular junction. Pretty good? All right, so again, we kind of learned this in a very compartmentalized way. So if you are missing some steps or you can't like go from one scenario to the next, just let me know and I'll try to smooth it out. Okay, so what happens when these action potentials here are initiated? I'm going to skip down to, don't worry about drugs and how it may alter this step, okay? That was just kind of giving you more information about what drugs could alter the chain of events that I'm talking about here. So I'm just going to make it simple. We're just going to go to action potentials now that propagate down these T tubules. Okay, so in this case, here This is essentially the same one. This is the diagram in your textbook. But it really doesn't show the neuromuscular junction, so I'm going to take it from here, okay? So we've already talked about the neuromuscular junction. Now we're right about here. And action potentials propagate along the sarcolemma down the T-tubule, right? They actually take a 90-degree turn, dive deep into the interior of the muscle, and they propagate all along the T-tubule here, basically causing the membrane voltage to depolarize, which then triggers the activation of these voltage-gated, remember they're sensing the voltage, that voltage change from those action potentials, you know, diving into the interior of the muscle. This dihydropyridine receptor is a voltage-gated calcium channel. So again, it can sense the voltage change. And the dihydropyridine receptor then goes through its own conformational change. It changes its shape, which you can see here is mechanically coupled. It's actually touching the ryanodine receptors that are expressed in the SR, the sarcoplasmic reticulum. Okay? So the dihydropyridine receptor is the one that senses the change in voltage in the T-tubule. There's a conformational change of this channel, and it causes the ryanodine receptor here in orange to open. Okay? And then calcium just floods. It actually just pours out of the sarcoplasmic reticulum into the sarcoplasm, into the intracellular space. Okay? Anybody remember the disease that's associated with mutation of the ryanodine receptor? Where it never closes. Wasn't myasthenia gravis, but that's a good guess. Myasthenia gravis is antibodies that attack the nicotinic acetylcholine receptor there, causing muscle weakness. This actually was, if there's a mutation of the ryanodine receptors and it just is continually open, this is what happened when patients are in surgery and they're given an anesthetic that keeps the ryanodine receptor open, and during surgery they just stiffen up, right? Malignant hyperthermia, right? So just remember, muscles, when they contract, they produce heat. So if you're in surgery and you're in a contractile state, your temperature is going to rise quite a bit. And it can become lethal. You can get to the point where your temperature rises to 106, 107 degrees. It's called malignant hyperthermia, and it's due to a mutation of this ryanodine receptor where it doesn't close when you're given a specific anesthetic, like halothane. They don't give halothane anymore during surgery just because of malignant hyperthermia. Okay, so is this blue or black? That's the voltage-gated calcium channel called the dihydropyridine receptor, and then you can see here, this orange one is the ryanodine calcium channel. Okay, so now what happens? Calcium floods into the intracellular space. That's what these two diagrams are showing you. Right? The red dots here are calcium. They literally flood into the intracellular space. We're like right here. And you can see that calcium binds to a couple of proteins that are on the actin filament. They're called troponin, troponin C. C is for calcium. So I'm going to take it from this diagram right here. This lower diagram is when you get calcium is resequestered by the Circa pump here. So we're not quite to this bottom diagram. We're actually right here, but I'm going to go through cross-bridge cycling first. Everybody with me? Am I going too fast? Yes?
Is the Circa pump resequestering calcium what ends this process?
Perfect. Yes. So the Circa pump is what ends the contraction, right? Because when you resequester calcium, it can't bind to troponin C. Cross-bridge cycling can't occur. Yes?
Circa pump is the one that causes rigor mortis, right?
Yeah, no. It's part of the cross-bridge cycling.
Okay.
Yeah. So you actually have the myosin head uses ATP.
Oh.
And so when ATP isn't around, because you're dead, that's what causes rigor mortis, yes. ATP usually leaks out of the cell, yes.
So how is like acetylcholinesterase involved in this?
Good point too. Okay, so that also is involved in relaxation. Okay, so imagine we go back to the neuromuscular junction. And there is an enzyme, this is a really good point, so thanks for bringing me back. Acetylcholinesterase breaks down acetylcholine. So obviously that will shut down the nicotinic acetylcholine receptors. You won't get action potentials. It also is involved in relaxation, yes. Yep. In addition to the Circa pump that causes resequestering of the calcium ions. Okay, great questions, you guys. All right, so let's go to the cross-bridge cycling. Is everyone actually feel pretty comfortable with the anatomy of the myofibrils? Yes.
Oh, we had to, sorry, go back.
Oh, that's okay.
Two proteins, one of us troponin.
Yep. So that's where I was going to go with this one.
Okay.
Yep. So here's the diagram for that.
Oh.
Yep. So you can actually see this is how troponin and tropomyosin are organized. I'll go back to how the myofibrils are organized in just a second. So calcium, you can see here, binds directly to troponin C, right? It's easy to remember that because the C stands for calcium. So you can see this troponin right here is also closely associated with troponin T and troponin I. Troponin T binds troponin with tropomyosin, okay? Troponin I binds the actin molecule and troponin C. So the three basically subunits that make up troponin are troponin I, troponin C, and troponin T. And you can see that troponin actually binds to tropomyosin all along the actin filament, about 40 nanometers. There's a troponin molecule every 40 nanometers along the actin molecule. So I don't know if I mentioned this in class, but it's kind of interesting. Troponin is associated with cardiac muscle as well, right? Because cardiac muscle also have actin and myosin, and they're organized in sarcomeres, they're striated. So when you actually go through a heart attack, right, myocardial infarction, one of the markers, what happens with heart attack is you don't get blood supply to certain parts of your heart. So it causes what's called ischemia. Cells die, and all of the inside parts of the cell just get released into the bloodstream. So one of the markers that doctors look for if they suspect a heart attack is troponin in your bloodstream, because that's what's you're going to release the troponin and all of, like I said, all of the other kind of markers, but they specifically look for troponin in your bloodstream if they suspect that you've had a heart attack. Interesting, right? Okay, so cross-bridge cycling. Here's what happens. I don't know if you can kind of follow along with this particular diagram, but with calcium, remember now there's a huge influx of calcium into the intracellular space. Calcium can now bind with troponin C, and what it does is it actually moves that tropomyosin out of the way, revealing these black dots, right? That's the actin and myosin binding sites. It reveals the binding sites, and now actin and myosin can interact with each other and cross-bridge cycling can occur. Okay, so you can kind of see that with this diagram. With calcium on board, you can see that the troponin actually moves the tropomyosin out of the way, revealing these binding sites. All right, so this is the diagram to kind of, think about when you're trying to memorize or, really know what cross-bridge cycling is all about. You can see here, this is where we would start. Calcium rises. You actually now are seeing that myosin head. Now, ATP has already been hydrolyzed to ADP and inorganic phosphate. Can you see that pretty well up there? ADP and the inorganic phosphate. So it's already been hydrolyzed or broken down into ADP and pi. And as soon as both of these molecules are released from the myosin head, that's what causes the power stroke. Okay, so it starts off a 90-degree angle. On the myosin head, you have ADP and pi. As soon as those are released, that causes the power stroke, moving those Z lines, the Z disc, closer together. Okay? Now, you can see that the myosin head and the actin molecule are still interacting with each other. In order for them to release from each other, ATP actually has to bind to the myosin head. That releases the myosin head from the actin molecule. Does that make sense, you guys? I can guarantee this is going to be one test question. What's responsible for the power stroke? What's responsible for the release of the myosin head from the actin molecule? Okay. ATP has to be around to release the myosin head from the actin molecule. And then as soon as the ATP molecule is hydrolyzed or broken down to ADP and pi, it re-energizes, right? It re-energizes it, brings it back to 90 degrees, allows for actin and myosin to bind again to each other. Okay? So it's just really 3 steps if you think about it that way. There's a little more to that, but this has been simplified so that there's only really 3 steps. In a 90-degree angle, you have ADP and pi that are attached to the myosin head. As soon as those 2 molecules are released, that's what's responsible for the power stroke. Then you need ATP to bind for the myosin head to be released from the actin molecule. And then when ATP is hydrolyzed or broken down to ADP and pi, it re-energizes the bridge, and it can, right? And so it just literally just ratchets, bringing those Z discs closer and closer together. Yes?
Just to confirm, the binding of actin triggers the release of ATP hydrolysis, which is the power stroke?
Yeah, I would say the binding of the myosin head to the actin filament eventually leads to the release of ADP and pi, which is responsible, the release of ADP and pi is responsible for the power stroke. But at that point, the myosin head is still attached to the actin molecule. You need to have ATP attached to the myosin head in order to release it from the actin filament. And then when ATP is broken down to ADP and pi, that re-energizes the bridge and allows for it to associate with the actin filament again. So if there's no ATP around, right, with death, that's what causes rigor mortis, right? No, it's just going to be pretty simple. It's just going to be, I mean, over each semester it's a little bit different, but it's really what is responsible for the release of the myosin head from the actin filament. What's responsible for the power stroke, right? It's just going to be something easy like that with 4 different foils. It's all multiple choice. Pretty good, you guys? All right. If you need some text, this is a nice slide to go through. It kind of gives you an idea of these different steps. Okay, now you can actually see how the myofibril, remember, this is a myofibril within a muscle cell, how this interacts with the actin filaments. So the myosin head, you can see, is actually ratcheting along that actin filament, bringing this Z line, Z disc, in this direction. Oops. Whereas with this Z line, it brings it in the opposite direction towards that M line. So the Z discs are going to get closer and closer together. Okay, so what happens? This is kind of a nice slide because you don't know. The next slide kind of tells you what happens. What happens to the A band? Remember, the A band always remains unchanged. The A band is technically from the myosin head to the myosin head. It's this darker shaded area right here. And it always remains unchanged. The I band here is going to get smaller and smaller as contraction occurs. The H zone also gets smaller and smaller. And the 2 Z lines move closer and closer together. But the A band remains unchanged. So this next slide really kind of hits home, right? You can see how when contraction occurs, A bands stay exactly the same, I band is reduced, H zone is reduced, and the Z lines get closer and closer together. So, relaxation, how does that occur? Acetylcholine esterase breaks down acetylcholine in the synaptic cleft, and the circuit pump resequesters the calcium back into the SR. What does that do? When calcium is resequestered, it can't bind to troponin C. Tropomyosin then moves back, occluding or blocking the actin binding sites, and the muscle slides back to its original position. Okay? Pretty good? Yeah. Acetylcholine esterase, is the enzyme that breaks down acetylcholine in the synaptic cleft. Yeah.
So that's like holding contraction. If I like squeeze my hand and let go.
Yeah.
So what if I'm just like squeezing my hand and holding it?
Some of your muscles will start to fatigue, and then other muscles will actually take over until your whole muscle finally fatigues if you keep that grip. So we'll get into that in a little more detail. You've got the slow, the fast twitch muscles, the slow twitch muscles. Some fatigue at different rates. And then you recruit other muscles to try to keep the same strength. Yeah. Does that make sense?
Yeah.
Okay, yeah. Until you're just completely fatigued and then you have to drop it, right? Okay. All right, so I'm going to go back because I want to make sure that you guys understand the myofibrils. Okay. So the myofibrils are organelles inside the muscle cell, right? I'll take you all the way back to here. Right? These structures right here. Okay, this whole thing is a muscle fiber. and that is the muscle cell. This is also known as a myocyte. Myo meaning muscle, cyte meaning cell. So, muscle cell is equal to muscle fiber is equal to myocyte. That is not the same as a myofibril. I'm showing you with my arrow here. This structure inside the muscle cell is a myofibril. And that's an organelle inside the muscle cell where all the actin and myosin are. Okay, all the actin and myosin. So I can just show you from this one, you can see the Z lines right here. From Z line to Z line, that's the single unit of contraction. That's called the sarcomere. Technically, it's from Z line to Z line. That's what a sarcomere is. Okay? Then you have this whole area right here is the A band. This area right here is the I band. And this reddish area, just right in between here, is the H zone. Okay? All right, pretty good, you guys? Let me just make sure that you guys know the anatomy of the myosin head here. So remember, this is the myosin. It has the actin binding site and the ATP binding site on the head region. This neck region, these are called myosin light chains. There's a lot of regulation that occurs, especially in smooth muscle. Remember the myosin light chain kinase? It actually phosphorylates these light chains in smooth muscle. And then here's the myosin tail. Actin, you've already kind of seen. These are globular actin that come together to form a filament. Each one of these are globular actin, and they come together to form F-actin, which is filamentous actin. And then you have the troponin and the tropomyosin. Okay, pretty good? All right, that's pretty much it. Okay, so if you go through that study guide, you'll notice that that's the sequence of events from excitation, you have the excitation of the motor neuron, all the way through to contraction of that muscle. Okay, pretty good? All right, so Lecture 8, Skeletal Muscle Dynamics. Now, the main thing that I wanted you to get out of lecture relationships, and there's at the very end, the difference between skeletal muscle, cardiac muscle, and smooth muscle. That's kind of at the end of lecture 8, but it puts everything together right before we go into cardiovascular. Okay, so there were some important terms that I needed you guys to know. This was just showing you what happens with relaxation. Calcium has to be resequestered. Okay. All right, two terms that are really important, isometric and isotonic contraction. That should be on your definition sheet as well. Isometric means same length. Metric is about distance. Isometric, same length. In this case, I'm just going to walk up to the desk here. If I have my arm at a 90-degree angle and I try to lift this desk because I'm not strong enough, my muscles are contracting. If I had a force transducer on this table, it would measure an increase in force. But I'm not able to lift the desk, right? So this would be a typical isometric contraction because I'm not allowing the muscle to shorten. There's no bones moving, right? Isometric. Now, if I had a 10-pound weight here, and I was able to lift it up and put it down, right? These are isotonic contractions. Isotonic, tonic refers to the strength. And so the weight of this book bag isn't changing at all. And I can put it down and lift it up and put it down and lift it it up, and I'm allowing the muscle to shorten, bone is moving, and I'm lifting the same weight over and over again, isotonic. Okay? So isotonic, you can kind of equate that with strength training. Isometric is more like what? Core endurance, core strength training, when, like Pilates, where you're not really moving bone or anything. Any questions, you guys? I hope I didn't just confuse you. Yes?
There's a question on the outlines I was confused about. Is an isovolumetric contraction isotonic or isometric?
It is isometric. Okay, so that's a really good question. When you're thinking about the heart, and this is during the phase where the ventricles are starting to contract, but the pressure inside the ventricles hasn't exceeded the pressure in the aorta. So it can't, those semilunar valves aren't open yet. So it's generating pressure. The pressure inside the ventricles are getting higher and higher and higher. But at that point, the muscle's not allowed to shorten. Okay, so it is an isovolumetric, same volume, or an isometric contraction. It's not until the pressure in the ventricle exceeds the pressure in the aorta that semilunar valve opens up, and then the ejection phase. Now the muscle's allowed to shorten, and the blood's allowed to be ejected out. Yeah, that's a good question. Okay, so just need to make sure that you guys understand that there's an what the difference between isotonic and isometric contraction is. Now, when you're taking a look at any muscle, you're going to have parts of the muscle that have the actin and myosin, and those are the active elements, or what's called the contractile elements. But within the muscle, and right outside, like where the tendons are, there are elements that are like rubber bands. They're elastic elements, and they're more passive. So take a look at this too. You can see this titin here, now that you know the structure a little bit better. This titin is also considered an elastic element. It's like a spring, and it kind of holds all of this in place. Okay? So we have an isometric contraction where the muscle's not allowed to shorten, it stays the same length. Isotonic concentric contraction is where the muscle's allowed to shorten. And an isotonic eccentric contraction, where the muscle actually lengthens because that afterload, that weight is so heavy that it actually lengthens out the muscle. Okay, so we went through the different relationships, and you need to know the 4 different relationships. Yes?
Really quick, what would be an example of something that happens to make a muscle shorten, and what would make a muscle lengthen?
Yes. So a typical isotonic concentric contraction is where you're actually picking up a 10-pound weight, and that's strength training, where you're moving the bone, and you're actually picking up the weight, and the muscle is shortening. Now, they say that when you put down a heavy weight, a lot of times if you do it right, it lengthens the muscles, and that would be an eccentric contraction. Sometimes, if you do it right, it's supposed to cause these tiny little micro tears, which build up myofibrils in each muscle cell, which causes your muscle to get bigger and bigger. So it's actually the eccentric, the eccentric part of it that causes the micro tears, where you really build up muscle. All right. So for each one of these relationships, I want you to understand the relationship, but also the molecular mechanism. So somebody asked me about what is the molecular mechanism for each relationship, and I'll make sure that's kind of what's happening at the cellular level, okay? And I'll make sure that you understand that. So let's just go through the intensity force relationship. That's the top figure here. And if you take a look at this diagram, what it is a whole muscle that's in this bath. It's being bathed with nutrients and oxygen to keep it alive. And you have a stimulator that's going to give it a little bit of a shock, and that's going to cause contraction of the muscle. But in this case, this is an isometric contraction, so we're not allowing the muscle to shorten. We're just going to measure the force generated. Okay? So on the y-axis, this is actually in newtons, or a measurement of force. Okay? This would be in time. And you can just send it a little bit of a shock. So if you guys were in lab that one day, we actually had electrode that passed a current that gave you a little shock and made your thumb twitch. And we could ramp it up, right? We could go from 2 micronewtons to, I think it was, what were the units of that? Amps. 2 amps to microamps to 20. So essentially what you're doing is, when you send a small shock to this whole muscle, you might get a little bit of force generated, and then you allow the muscle to relax, and it goes back down to its original value. This would be called a single twitch. So this is just single twitch responses. Then the black bar, you're actually passing more current, giving it a greater shock, and you can see it actually increases the force even more until you get to a maximum, right? This would be a maximum stimulus intensity. The stimulus is the shock with a single twitch, and then it doesn't matter if you keep increasing the shock, you've already maximized the amount of force that can be generated with a single twitch. Okay? So this is called the intensity-force relationship, and what's happening. I want you to have a mental picture so that you can really understand what's happening. When you give it a little bit of a shock, you're only recruiting a couple of motor units or muscle fibers in that whole muscle. So you can only generate a little bit of force. But as you increase the shock, you recruit more and more muscle fibers until finally you've recruited all of the muscle fibers in that whole muscle. So even if you increase the shock even more, you've already recruited all the muscle fibers that can be recruited. Okay? Pretty good? Okay, so what is the frequency-force relationship? Now we're talking about the frequency, the number of times that you shock that particular whole muscle. Okay? So in this case, we've decided we're going to use a maximum stimulus. We've already determined what that maximum stimulus is. So we already know with this stimulus, with this amount of shock, we're recruiting every single muscle fiber in this whole muscle. But now we're going to increase the frequency. Now you can see this isn't just a single twitch. We can shock it and shock it again really quickly and get summation and eventually a new plateau, a new saturation point at that particular frequency, and that's what's called tetanus. Okay, so tetanus is technically the maximum amount of force that can be generated using a maximum stimulus intensity at a specific frequency. You want me to say that again? It's the maximum, tetanus is the maximum amount of force that can be generated with a maximum stimulus intensity at a particular frequency. Okay? So here's the formal definition of summation and tetanus. And you can see that we're reaching tetanus at different frequencies. So I want to make sure that you guys understand this too. you can reach tetanus at this particular frequency. This would be 100 milliseconds every time. This is the firing rate of 100 milliseconds, or 1, 2, 3, 4, 5 shocks per 500, 400 milliseconds. And at this frequency, you can see we reach tetanus, but it's an unfused tetanus. Now we can really ramp up the frequency, and you can see this generates a lot more force. And this is what would be called fused tetanus, because now you can't actually see the relaxation events. Okay. You guys pretty good with that? This is called the frequency force relationship. Yeah.
So when it becomes like fused tetanus, is that just when the muscle like won't relax?
Yeah.
Basically, or is
Yeah.
that like
I would say physiologically, it's like having a cramp.
okay?
Yeah, that's what happens. And it's usually because you're dehydrated and your motor neuron is firing at such a high rate that it just is cramping up that whole muscle. Now, the reason why the refractory period in the ventricular cardiac action potential is so long is it's because it prevents summation and prevents tetanus, because that's the last thing you'd want is your heart to cramp up, right? That would be really bad. All right, so now you know a little bit about summation and tetanus. I think those are on your definition sheets as well. And what is the molecular basis? Okay, so what is the molecular basis? It's not about calcium. This figure right here is telling you that it's not about calcium. This shows you that with a single twitch or with multiple stimulation where you get summation and tetanus, calcium remains the same. There's no increase in the amount of calcium in the intracellular space. And you can see the contractile force, the contraction force, is really much higher with multiple stimulation than with a single twitch. But it's not about calcium. It's about these elastic elements. So I want you to think about these elastic components like titin as shock absorbers. Okay? So at first, when you stimulate the muscle, you have a lot of shock absorber that are taking up a lot of the force. Okay? They're absorbing a lot of that force from the shock. But as you continue to increase the frequency of that shock, you can see it tightens up the elastic elements. And so when you finally reach tetanus and you give it a shock, all of the force can be translated to the ends of the muscle, and you can measure a maximum force generation. There's no more shock absorbers to absorb that force, okay? So it's about tightening up those elastic elements until you reach tetanus and you finally give that final shock, and then all of the force can be expressed at the ends of the muscle, okay? Does that make sense, you guys? You're basically reducing the shock absorber when you tighten up those elastic elements. Okay, so let's go on to the length-tension relationship. I want to finish up with skeletal muscle soon here so we can get to cardiovascular. All right, so length tension is all about what's called preload. Length tension is about preload. It's about setting the length of the muscle before stimulation. Okay? So you naturally have an when you're talking about skeletal muscle, you have a natural optimal length. And luckily our muscles are already attached to our bones at optimal length. Okay? So that's what we're talking about right here. Let's go to, it's basically the middle muscle right here. number 3, where you can see at optimal length, we have the maximum amount of myosin and actin interactions. And then you stimulate the muscle, you're going to get the maximum force generated with a single twitch. If the muscle is too short, you can see some of the actin overlaps and there's really nowhere to go. And then this is a really short muscle where you're not going to generate very much force at all. You can actually see even the myosin heads are bunched up against the Z disc there. If the muscle's too long, you can see that the myosin heads have slid past the actin molecule and there's not many actin and myosin interactions. So you're not going to generate as much force as well. Okay, so this is what the graph looks like. You do have an so the y-axis is force, okay, and the x-axis is the muscle length. So at LO, that's when you can generate the most force. If it's too short, it really falls off. If it's too long, it also falls off. Okay, pretty good? Now, remember there's these elastic elements that also play a role. So that's the red line here. When you stretch that muscle out, there are those elastic elements, the titin and the tendons, that are like rubber bands. They're going to try to contract and recoil. So that's what this red line is, the passive elements. So when you actually are looking at it in the laboratory, it's the phenomenon. What you observe is the black line, right, which is the addition of the red and the blue lines. The blue line is the passive elements. And so if you really stretch out the muscle, you're going to get that recoil effect due to those elastic elements here. All right, so that's also known as preload. And in the heart, remember it's the amount of blood that's returned to the heart, the end diastolic volume. That's setting the length of the muscle, right? That's called the Starling mechanism. Frank Starling mechanism. And that has everything to do with length-tension relationship. It's setting the length of the muscle before stimulation. Any questions, you guys? All right, load velocity is the only relationship that is looking at examining an isotonic relationship. Okay, isotonic relationship. So in this case, you're actually looking at different afterloads and how that changes the speed of contraction. Does that make sense? You're basically hanging different muscles, right? Different muscles here. And this isn't a good diagram because you're actually trying to pick up different weights. And as you increase the weights, a light load, intermediate load, and heavy load, it actually changes the velocity of contraction. So what is velocity? It is distance over time. So all you would do is take the slope of these different curves, and you could generate this particular graph. This would be what happens when you have a light load at the end of a muscle, and you stimulate it to contract. It will contract and then relax. If you put a heavier load on there, it will contract and relax. And then a very heavy load, you can see it just kind of contracts just a little bit and then relaxes. So to take the velocity, you would have to take the distance over time. You want the slope. So the slope here is pretty steep, right, for the light load. And if we actually generated the graph, it would be this green dot right here, just below the maximum shortening velocity when there's no load. Then if we go back, the slope of the orange one is right here. We'll plot it right there. And the slope of the purple one right here is there, okay? You can actually generate a load velocity curve by doing these experiments. This is actually where the weight is so heavy that it doesn't allow for muscle contraction. What would that be? If the weight that we hang on that muscle is so heavy, it doesn't allow for muscle contraction. That'd be isometric, right? The maximum isometric tension. And then if the load gets even more heavy, it'll start to lengthen the muscle. So that's a typical load velocity curve. The speed of shortening decreases as the load increases. All right, you don't need to know anything about this graph except for what is preload and what is afterload. Okay, so this is a very contrived experiment. It probably would never work in real life. But it's a nice diagram to kind of tell you what preload and afterload is. Preload is, in this case, it's a weight that's setting the length of the muscle before stimulation. Preload. And when you stimulate it, it's like a teeter-totter. It's going to pick up this afterload. So the afterload is anything that this muscle has to work against. The heavier the afterload, the harder this muscle has to work to pick it up. All right? So in the heart, the afterload is blood pressure. If you have super high blood pressure, then your heart has to work harder to get the same amount of blood out. Okay? So afterload in the heart is total peripheral resistance. It's the resistance in the systemic system. If someone has high unchecked blood pressure, they'll go into the hospital and they'll image their heart. Their heart will be huge. It's called hypertrophy of the heart. Because just like any muscle, if a muscle has to work hard against high blood pressure, it's going to get bigger and bigger and bigger, just like if you were strength training, right? And that's a good sign of heart disease, is when you have hypertrophy of the heart. Okay? All right, ATP. With ATP, I do want you guys to know what fatigue is. Fatigue is when basically you deplete the cells of ATP, and the potassium gradient changes as well, because a lot of potassium floods out of the cell, especially into the T tubules with contraction, prolonged contraction, all contributing to fatigue. And then know the difference between slow, oxidative, fast, glycolytic. Don't worry about the intermediate one, just the two extremes. Okay, don't worry about that one. This is a nice slide because it really highlights the differences between the fast glycolytic and the slow oxidative. Fast glycolytic is here in light orange. Slow oxidative is the red ones here. So without even looking at a chart, I can tell you that the fast glycolytic are larger because they have more myofibrils. They're more powerful. They're the muscles that you use for sprinting and jumping. Okay? So they have more myofibrils within each muscle cell. But you can see that they don't have a very rich blood supply. These black dots are the blood supply. So they don't have a rich blood supply. They don't have much myoglobin, which is a molecule that binds oxygen. That's why they're considered the white meat, right? White meat. And so they fatigue really easily. Remember the secret to crocodile wrestling. Made up of all white meat. So, no rich blood supply, not a lot of myoglobin, not a lot of mitochondria. There's just not a lot of opportunity to generate ATP. So they fatigue easily. The slow oxidative, like this red one here, has a very rich blood supply. You can see all those black dots. So they're constantly being perfused oxygen. They have lots of myoglobin. That's why they're red in color. They're rich in mitochondria, but they're not as powerful. But they don't fatigue, which is great. It's like the muscles in your back, your posture. And if you need a chart to kind of look that over, you can look it over with this one. It's a good one. All right, I won't say don't even worry about any of these slides through here. But do know the difference between smooth muscle, cardiac muscle, and skeletal muscle. The major one that I want you to know is this one. Okay, this is smooth muscle. All three of them depend on an increase in intracellular calcium. But with smooth muscle, it activates an enzyme called calmodulin first. Then calmodulin activates myosin light chain kinase. Remember, that's on the neck of the myosin head. So when you activate myosin light chain kinase, it phosphorylates the neck of the myosin head, allowing for myosin and actin to interact and causing those cross-bridge cycling so that you can get contraction. So when calcium is resequestered or removed from the cell, the myosin light chain kinase is inactivated, and this myosin light chain phosphatase takes over. A phosphatase is anything that dephosphorylates, takes off these phosphate groups. So it dephosphorylates that myosin head there, that neck, and now the myosin and actin can't bind. So you don't get cross-bridge cycling, and you get relaxation. Okay, I think that's pretty good. I think the only other terms for this one is myogenic versus neurogenic. Myogenic is referring to cardiac muscle. Oh yeah, you have a question.
So for the smooth muscle, is the phosphatase or whatever kind of like the circa pump or skeletal? Or
Yeah,
is that what ends the cycle?
That's a really good question. There are, and you don't see it in that diagram, there are pumps. This is a plasma membrane calcium pump that could remove calcium from the cell. Or sometimes smooth muscle cells do have intracellular stores. So you may have a circa pump that's also resequestering calcium that you just don't see here. But it is responsible for relaxation along with the phosphatase that dephosphorylates that myosin neck region. Okay? Yeah, good question. Both of those contribute to relaxation in smooth muscle. And remember, there's two types of smooth muscle. One that acts in coordination together. They have gap junctions, so it acts as a single unit. It's called single unit smooth muscle. Okay? And then some that are multi-unit. They don't have gap junctions, and they work independently of each other. All right, so myogenic versus neurogenic. In cardiac muscle, because of those specialized SA nodal cells, they depolarize all on their own, and that depolarization is what's responsible for the excitation and contraction of cardiac muscle. So it's inherent within the muscle cells. That's why it's called myogenic. Myo refers to muscle. Genic refers to origin, like genesis, right? So myogenic means that that electrical signal starts within the muscle cell itself. Neurogenic, like with skeletal muscle, means that the electrical signal has to come from a neuron, a motor neuron. Neurogenic. And that's excited by neurotransmitters. There is a real difference, again, between the dihydropyridine and ryanodine interactions, whether we're talking about cardiac muscle or skeletal muscle. Skeletal muscle, they're physically interacting, and most of the calcium, 95%, comes from internal stores. So it really doesn't, the dihydropyridine receptors don't conduct calcium, they just physically interact with the ryanodine receptors, which opens them and allows for calcium to flood into the intracellular space. But with cardiac muscle, you can see that they're actually not physically interacting. Calcium flows through the voltage-gated calcium channel called the dihydropyridine receptor. And that calcium actually activates ryanodine receptors, which is a calcium channel as well, and releases calcium from internal stores. This is called calcium-induced calcium release. So there's just a little bit of a difference in the morphology between cardiac muscle and skeletal muscle. And major difference too is that cardiac muscle has that intercalated discs that have all the gap junctions, so that that electrical current can go from cell to cell to cell pretty easily. Okay, so let's go through cardiac muscle. And I'm just going to hit the highlights here. I would say that much of the introduction is not going to be on your exam. You will have to know the anatomy of a four-chambered heart. So the four chambers, this is typical of mammalian organisms, whether we're talking about mammals or humans, okay? They have four-chambered hearts, and crocodiles, and birds, which is kind of interesting. Crocodiles are more, now that they've analyzed the DNA, are more closely related to birds than they are reptiles. So they're actually thought to be in their own kind of branch now, crocodiles are. So they have four-chambered heart like birds do. Cool, right? Okay, so be able to label this. And I would know something about the pericardium. The outer membrane is the parietal pericardium. Then there's a fluid-filled interior. And then the membrane that's closely associated with the heart muscle, the myocardium, is called the visceral pericardium. Okay? And then the interior here are lined with epithelial cells. But I'm going to go ahead and just start with the electrical component here. All right. If you guys wouldn't mind getting out your cardiovascular essential calculation sheets too. Do you guys feel pretty good about that, or do you want me to go through some of those calculations? The calculations should be pretty trivial, but it's understanding them. So, I know people at home are going to hate me for this, but hopefully they can still hear me. The equations that are not going to be given to you are Ohm's law, good walking around knowledge, and cardiac output. So, just so you guys know, Ohm's law, mean arterial pressure is equal to cardiac output times total peripheral resistance. Right? How do you calculate cardiac output? Cardiac output is equal to heart rate times stroke volume. All right, how do you calculate to 1 beat every 0.8 milliseconds. Oh, let's say this is 800 milliseconds or 0.8 seconds. Makes more sense, right? So heart rate for every beat would be 0.8 seconds. And then we would just multiply that by 1 or 60 seconds over 1 minute, which is 1. That would give us a heart rate of 75 beats per minute. You guys with me on that? So, you would be able to calculate heart rate, 75 beats per minute, times stroke volume. Usually I would, let's see, I might give you ejection fraction, or I would probably just give you stroke volume. But you would have to know this equation, heart rate times stroke volume is cardiac output. Okay? So if we say stroke volume is about 70 mils per beat, then you would be able to calculate cardiac output. Now, I will give you how to calculate mean arterial pressure. Okay? So remember, mean arterial pressure is equal to the diastolic pressure plus 1/3 the systolic pressure minus the diastolic pressure. And that will be given to you. So, if you can calculate cardiac output and mean arterial pressure, you'll probably need to calculate total vertical resistance, which is basically mean arterial pressure divided by cardiac output. Just rearrange that equation. All right. Did I not get those back? Okay. Darn, I don't have them with me. If you want to walk over to my office later, or I do have an answer sheet that's posted on Moodle. So you might be able to take a look at that. Yeah. Sure. Okay, so let's go through the electrical currents really quick. Remember, they start in the SA node. And I do want you to know something, and I'm just going to make it pretty simple for you guys. I want you to know something about the action potential. Starts in the SA node. Anybody remember what phase 4, what channel is responsible for phase 4? This eroding depolarization that eventually reaches threshold, fires an action potential. This is actually your pacemaker current. It's the funny channels. These are, and I will say on the exam, HCN or funny channels. I just want you to identify it. If you were to take cell physiology with Dr. O'Grady, he would have you know that this is called the hyperpolarization cyclic nucleotide gated channels, and he'd have you know a lot more about this particular channel. I'm just calling them the funny channels. And on the exam, you'll see HCN or funny channels. Just be able to identify that this eroding depolarization, this phase 4, is due to those funny channels. And because those cells are expressing those channels, That's what's giving you the autorhythmicity, right? That's what's allowing your heart to beat on a regular basis. Those are your pacemaker cells, and if you weren't expressing those channels, then you wouldn't have that capability. Okay? Those are the pacemaker channels. Phase 0 is due to calcium channels, pulling the membrane voltage towards the equilibrium potential for calcium. Very depolarized. And then anything hyperpolarizing the cell, just know that's potassium. So phase 3 is due to the opening of potassium channels, bringing the membrane voltage back down to more hyperpolarized voltages. Okay, so we'll come back to this. All right, so the reason why the SA node are your pacemaker cells is only because they fire first. They're the fastest. So they depolarize this area, and then the depolarization wave, you can see here in yellow, sweeps through the atria, ends up at the AV node where it slows down quite a bit because there's not a lot of gap junctions in the AV node, allowing enough time for the atria to completely contract before the signal runs down the septum to the apex of the heart, up the Purkinje fibers here, and then propagates through the entire ventricular mass. And then relaxation occurs. Okay? So if you were looking at this, well, let's take it back to the action potentials within the ventricles. This is what the action potential in the ventricles look like. Remember, we have this very long action potential. The duration is very long. And that's, again, because the refractory period, it keeps it depolarized for a long period of time so that it stays in the refractory period. So you don't get summation, you don't get tetanus. It lasts as long as the contraction itself. Phase 0, what's that due to? Sodium. Yeah, I heard that. Sodium. This is different than the SA node. Phase 0 in the SA node is due to calcium. In the ventricles, in the ventricular myocyte, it is sodium. 1, even though this is just a little dip, it is potassium. 2 is due to calcium, that plateau phase. This is called the plateau phase. It's due to calcium. And then 3 and 4 are due to potassium. 3 and 4 are due to potassium channels. So why is this important? Why are you guys having to understand why these different channels open and close? Remember, it's the ion channels that are responsible for the action potential waveforms. And if you're talking to somebody who's a patient that has arrhythmias, they're taking medication that alter these different channels to change their action potential waveform so that they don't have arrhythmias. So there's different classes of antiarrhythmic drugs, which you would learn in like a vet school, right? If you're treating an animal even. The different types, like the class 1 are the sodium channel blockers. Class 3 are the calcium channel, verapamil, right? So if you're giving, patients these different antiarrhythmic drugs, they're targeting these ion channels, okay? All right, so those are action potentials. Now, I know I get a lot of confusion with what's the difference between an action potential and the EKG, all right? I get there's a lot of confusion between that. An action potential is when I take an electrode and I impale one cell within this zone, and I measure the membrane voltage changes in one cell. I can measure the action potential. But the EKG is looking at all the action potentials as they go from cell to cell to cell, right? Sweeping through the entire heart. So this is what it looks like. Sorry. Okay, you can kind of see all of the different action potentials and how they play a role in the EKG. Okay, so this, I'm making it simple again just for the exam. I do need you to know P wave is atrial depolarization. The QRS wave is ventricular depolarization. And then the T wave is ventricular repolarization. And the questions will be very simple. It's just going to be literally P wave is, and then you'll have different answers. A, atrial depolarization. B, right? It's going to be pretty simple. All right. So now, the end of this is actually how the autonomic nervous system plays a role in cardiac pacing. Okay? Now, there's a lot of information through here, but the main idea is really about G protein coupled receptors, okay? The sympathetic neurons and the parasympathetic neurons, they are literally innervating the SA node, and they're controlling the pacing of the heart. You know this because when you get frightened, your heart speeds up quite a bit, right? Ah, you startled me. Oh my gosh, my heart is racing. Ah, you guys know that. The parasympathetic does the opposite. It slows the heart down. So how does it do that? The sympathetic neurons, when they're activated, they release norepinephrine, right? Norepinephrine, which actually binds with adrenergic receptors, and they're GS coupled. GS coupled. So that increases cyclic AMP in the cell, which directly binds to the funny channels and alters this phase 4 slope. You can see it increases the slope, so it actually reaches threshold quicker, increasing your heart rate, all because of GS coupled receptors. So it makes sense that the parasympathetic neurons are activating those muscarinic acetylcholine receptors, which are GI coupled, right? GI couple is going to lower cyclic AMP in the cell, so you don't get binding of cyclic AMP to the funny channels. And it decreases its activity, making that phase 4 more shallow, so it takes longer to reach threshold. Kind of makes sense, right? Sympathetic GS, parasympathetic GI. The only addition here is those GERK channels opens up a potassium channel that depolarizes the cell right at the beginning of phase 4. They're called GERK channels. And if you, this is actually interesting. We never really ascribed a role for beta gamma, but when the GI coupled receptors are activated, it's the beta gamma that migrates over. Remember, the alpha migrates over to adenylyl cyclase and basically inhibits adenylyl cyclase. But the beta gamma in this role migrates over to that potassium channel and opens it up, hyperpolarizing the cell and making it more negative here at the beginning of phase 4, which further causes it, prolongs the time period before it reaches threshold. So, any questions, you guys, about the sympathetic and parasympathetic role in heart rate, cardiac pacing? Okay, let's go. Last lecture. Now, I know you guys have just done this one. So, hopefully it's recent enough. And I'm just going to hit the highlights. Oh, here's another. You guys remember, I threw that up there, but I didn't go through that. Systole is when the ventricles are completely contracted. Diastole is when the ventricles are completely relaxed. So there's a real pressure difference between these two events. This is the Wiggers diagram, and this really helps you kind of think through all of the different events with each heartbeat. I'm going to walk you through it. I think we still have some time. And then I just want you to listen carefully. I'll tell you some questions that I could ask you about these different events. One of them is, what valves are open and what valves are closed? Okay, so just remember the AV valves open and close at the same time. AV valves. That's the right and left AV valve, also called the tricuspid and the bicuspid, or the tricuspid and the mitral valve, right? And the semilunar valves open and close at the same time. All right, so let's go ahead and go through this. First thing that happens with every cardiac cycle is the electrical current sweeps through the atria. The P wave happens first. Then the atria contract, okay? Now the top one is showing you what the pressure changes in the left ventricle. The middle one here are the volume changes in the left ventricle, and then your heart sounds, which gives you an idea of what's happening with the valves, okay? So as soon as the P wave occurs, the electrical signal goes through the atria, you get contraction of the atria, and you can see there's a little bump in the pressure in both the ventricle and the atrium, right? You can see the atrium here in purple. So you get a little bump in pressure and a little bump in volume, blood volume in the left ventricle, okay? Then the QRS wave occurs. So the electrical signal now goes through the ventricle. And immediately what you'll notice is the ventricles start to contract, and as soon as the pressure in the ventricle, which is in the green, exceeds, right, gets higher than the pressure in the atrium. Imagine that in your mind. When the pressure in the ventricle exceeds the pressure in the atria, that's when those AV valves shut. Only pressure changes make those valves shut. And that would be responsible for this first heart sound. Boom, AV valves shut. Now, this whole area right here in green, right in between these two dashed lines, that's where the pressure in the ventricle is rising, 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 but look, there's no change in volume. This is an isovolumetric phase. Also, as we discussed earlier, an isometric change, an isometric phase. So, is there any blood being ejected at that time? That's always a good question. There is no blood being ejected. This is an isovolumetric phase. Iso meaning same, volumetric referring to the volume. So it's in between these two little dashed lines is the isovolumetric phase. The pressure is rising in the ventricles until you can see the pressure exceeds the pressure in the aorta. And that's when those semilunar valves open, and that's when you get ejection. See the volume now? Plummets. That's the ejection phase. So during the ejection phase, which valves are open? Semilunar valves are open, AV valves are closed. During the isovolumetric phase, right before that, which valves are open? None. They're all closed. Okay. Now, this space between these two little lines, this is the isovolumetric relaxation. This happens during the T wave, right? The T wave happens, the muscle starts to relax. As soon as the pressure in the ventricles falls below the pressure in the aorta, that's when those semilunar valves shut. So, what valves are open during the isovolumetric relaxation? Right in between these two lines. What valves are open during the isovolumetric relaxation? None. None. It's not until the pressure falls below the pressure in the atria. Now, which valves open? AV valves. Now you're going to get passive filling again between the atria and the ventricles. Okay? And then it starts all over again. Yeah?
So the ventricle pressure is just below the atrium?
Yes. Yeah. So blood's always going to flow from high pressure to low pressure. So you would have to deduce that the pressure in the atria is higher than the pressure in the ventricle. Yeah. Yes. Yes. So in the ejection phase, that is, if you can see the arrow right here, the ejection phase is between these two lines, right? So that's when the pressure in the ventricle exceeds the pressure in the aorta. It gets higher than the pressure in the aorta. Now it basically blows open the semilunar valves, and the blood is ejected from the ventricle into the aorta. And that's the ejection phase right here. So if I asked you a question, which valves are open, you would know that it's the semilunar valves. Yep. Yep. Yeah.
So is it between the two sets of dotted lines that all the valves are closed?
I would say the valves are closed in between this area right here, that tiny area, and all the valves are closed in between this tiny area. Yeah. Yes.
So the heart valves at the bottom.
Yep.
The first one would be the closing of the AV.
Yes.
The second one's the center.
Yes. Yep. Perfect. See, now you're starting to think about this chart, this diagram in a more sophisticated way. Yeah. Physiology is something you can't cram for, right? Imagine trying to cram all of this in a 2-hour period. You would say, oh yeah, I know that, and you just keep going, right? But you do have to think about it carefully, right? Pretty good, you guys? Okay, that's called the Wigger's diagram, and now it's much easier to go through the cardiac cycle, and you can see here's where the valves are open and closed. You'll be able to kind of go through that as well. It's much easier to go through this diagram now, knowing something about the Wigger's diagram. Okay, so you guys are pretty familiar now with all of this. There's a couple of additional terms. Bradycardia is an abnormal slowing of the heart. Abnormal slowing of the heart. It's usually diagnosed when a heartbeat is 40 beats per minute or lower. Bradycardia. Tachycardia is an abnormally high heart rate. It's usually diagnosed when the heart rate is 100 beats per minute or higher. So now if you guys are watching Grey's Anatomy or something like that, and they say, the patient's in tachycardia, VFib, now you know something about that. They actually do throw those terms out all the time. I'm like, oh, I see it on TV, and I'm like, oh, I hope my students saw that. All right, so you know that. Ejection fraction is the amount of blood that's ejected with every heartbeat. That would be stroke volume over end diastolic volume. And you can calculate stroke volume by just knowing something about end diastolic volume and end systolic volume. So this would be the amount of blood that's in the ventricle before contraction. And systolic volume would be the amount of blood in the ventricle after contraction, when the ventricle is completely contracted. And you can just subtract those to get stroke volume. All right, you guys, this is the Frank-Starling effect. This gives you an idea of as you increase the amount of blood that is forced into the ventricle, you can actually increase the stroke volume. You can get a more forceful contraction. Why is this? It's because you're stretching the ventricular muscle towards optimal length before it's stimulated. That's the length-tension relationship. I can guarantee you guys that will be a question. And it's always amazing how many people miss that question too. Just remember, this is called the Frank-Starling effect, or the Frank-Starling mechanism. Due to the length-tension relationship, allows the heart to automatically compensate for increases in the amount of blood returned to the heart. And then when you actually have a sympathetic surge, all of these different components are activated. Remember, it's a GS-coupled reaction. It activates pKa, which has effects on calcium channels, ryanodine receptors. This is the Circa pump. It actually increases the activity of the Circa pump. And it tightens up, tighten. So you get a more forceful contraction and an increase in stroke volume and a faster relaxation. So what you need to know is a sympathetic surge, you get frightened, is going to increase heart rate and stroke volume, which increases cardiac output. How does that affect mean arterial pressure? Goes up, right? Sweet. Yes. So if you increase heart rate and you increase stroke volume, you have to know that it's going to increase cardiac output. And when you increase cardiac output, even if total peripheral resistance stays the same, if you just plug it into the equation, you know that's going to increase mean arterial pressure.
You said it also increases the action.
It does. It actually causes a faster, if you look at that too, it causes a faster relaxation. See, this is the relaxation. And that's because it increases the ability of the cell to resequester calcium too. Yeah.
So then the parasympathetic is just the opposite of all of that, decreased heart rate, stroke,
Perfect.
cardiac output, and also
Yeah. Okay, so what is parasympathetic to? It decreases heart rate, decreases cardiac output. What would it do to GERK activity, that potassium channel? It would increase the GERK activity. What would it do to GI-coupled receptor activity? The GI couple, would it increase it? Yes. So just imagine just a list. And all you have to say, all the title says is, with parasympathetic stimulation, how does that affect cardiac output? How does that affect heart rate? How does that affect mean arterial pressure? All of that should decrease, right? But then there may be things that are thrown in like GERK activity, and that would increase. Adenylyl cyclase activity. that would decrease. Parasympathetic. Okay. So I think we can go through this pretty quickly and end here in about 15 minutes because we've gone through a lot of this before. I want to make sure that you guys understand Poiseuille's law. This is an equation that you don't need to run any numbers, but the whole idea here is this r to the 4th value, right? Any slight changes in the radius of a vessel is going to have huge changes on resistance. All right, so here's the equation. Like I said, I want to make this apparent for you guys. Okay, so Q is flow, right? So I think it's written this way. Change in pressure. Change in pressure is equal to Q times R. Ohm's law, right? This is just Ohm's law. This is flow, this is resistance, and this is the pressure gradient, which is the driving force. So, Ohm's law. The equation that they have right on the screen there is Q is equal to delta P over R, okay? And what they've done is they've defined R, right? So R happens to be equal to 8 Ln over pi R to the 4th. Yeah, good. All right, so R is equal to 8 times the length of the vessel, the viscosity of the blood, and then R to the 4th. That's the radius of the vessel to the 4th power. The tricky part about this is because R is in the denominator, they plug this in, so it's written like this. Delta P times pi R to the 4th over 8 Ln. Viscosity. See where I'm going with that? So, if I give you this equation, and I ask you something about resistance, you would have to mentally kind of flip this over to answer those questions. But it's a little bit intuitive, okay? So the longer the length of the vessel, the higher the resistance. The thicker the blood, the higher the viscosity, the higher the resistance. So even if you couldn't mentally rearrange this equation, I want you to intuitively know the resistance. The longer the length of the vessel, the higher the resistance. The higher the viscosity, the thickness of the blood, the higher the resistance. And the smaller the radius, the higher the resistance. So you should be able to intuitively answer any questions about resistance. All right. Know that the arteries are super elastic. When you push blood into the arteries, they're going to have a recoil effect to keep the blood moving even in diastole. It's very elastic, under very high pressure. And then as you get into the arterioles, that's where you can actually see that drop in pressure, because there's a lot of regulation associated with those arterioles. A lot of vasoconstriction and vasodilation in the arterioles, but you can see how that drops off dramatically. And then there's hardly any pressure in the veins. So if you nicked a vein, it's not as detrimental as if you nick an artery. I think I told that story about breaking an artery in my head when I was a kid. Man, there was a lot of blood. Yeah. Still scarred my sister. She still talks about that when we were kids. So, now, when you're talking about the capillaries, just know because the area of all the capillaries increases dramatically because it branches into so many different vessels, the velocity has an inverse relationship with the area. So it causes the blood velocity to slow down quite a bit through the capillaries. Now, to answer that question, I want you to have a mental picture of that Mississippi River. At the widest points, the water is rushing by you. I mean, it is, if you jumped in the water, you would be caught up in the speed of that water. But as it branches into tinier and tinier little streams and brooks, the velocity of the flow slows down quite a bit because the surface area, as it branches like a delta, increases the area, so it causes the velocity to really decrease. I wouldn't worry about any of this, the modeling of circulatory systems. But, and the sphygmomanometer isn't going to be on the exam either, so don't worry about that. Arterioles are a dynamic site of regulation, and it's all about vasoconstriction and vasodilation. The next thing that's really important, I would say know the difference between intrinsic mechanisms. Remember, I just said I wanted you to know intrinsic versus extrinsic. Intrinsic are these local controls. It's basically maybe oxygen or carbon dioxide can have some effect, like the environment that the smooth muscle is in is going to have an effect on vasoconstriction or vasodilation. Those are intrinsic controls. Extrinsic controls are just hormones or neurons. Outside controls. And again, it's the arterioles where there's a dynamic site of regulation. Capillaries, the main thing with capillaries is the net filtration pressure. This is where we started on Monday. So, know the difference between filtration and absorption, and know how to calculate net filtration pressure and what it means. Okay? So net filtration pressure is the net hydrostatic pressure minus the net oncotic pressure. So net hydrostatic pressure is the hydrostatic pressure in the capillaries minus the hydrostatic pressure in the interstitial fluid. Okay, so you might be given a graph like this that gives you the information about pressure in the capillaries and oncotic pressure in the capillaries. And then I would give you specifically the hydrostatic pressure in the interstitial fluid, which is close to 0. It's usually about 2.
And then where will we be given the equation?
Yes, you will be given the equation. It's actually written in the question itself. Yeah. Okay, so again, the calculation is trivial though, right? You guys know how to calculate. You should be able to plug it in. But what does it mean? If it's a positive number, what does that mean? Filtration, yes. It's going out. Fluid is being forced out of the vessel. If it's a negative number, absorption. Fluid is going to be forced into the vessel. And when you're talking about a typical capillary, all of the fluid that's forced out, only about 9/10 of the fluid is returned. There's still 1/10 of the fluid that remains in the interstitial fluid. That's also called edema. If it accumulates in the tissue, it's called edema. So how is that extra fluid, that last 1/10, returned back to the circulatory system? Lymph system. That is one of the major roles of the lymph system, is to return fluid, not blood proteins. It's the fluid that's returned back to the circulatory system. So, good example is filarial worms and elephantiasis. If you block the lymph system, you're going to get profound edema in the extremities. Or if someone you know has their lymph nodes removed, right, that could cause severe edema in the arms, in the extremities. Okay, so good examples, illustrations, why that's important. Veins act as a volume reservoir. Remember, they have one-way valves. They have very compliant walls. What does compliance mean? Compliance is giving you some information about the relationship between volume and pressure. So if something is highly compliant, it means just a little bit of pressure translates to huge changes in volume. Okay, that's compliance. So because of that, veins hold about 60% of the blood, and you have to use other muscles like your quad muscles or your respiratory muscles to return blood back to the heart. Last thing, you guys, baroreceptors. There are a few questions on baroreceptors. This is again telling you something about sympathetic and parasympathetic nervous system. Okay? Let's just say you're severely dehydrated and you don't have much blood volume. Has anybody ever noticed this before? You might have noticed this in the wintertime. What's that going to do to those baroreceptors? Let's take a look at this. What would you suspect? Is your mean arterial pressure going to fall or get higher? If you're severely dehydrated and you have hardly any liquid in your body, you're going to have low blood pressure, right? You're not drinking enough water. Your blood pressure falls. What is your baroreceptor firing rate going to do? Well, here's the mean arterial pressure. It's going to fall, right? If you go along this line, baroreceptor firing rate is going to fall. That sends a signal to your cardiovascular control center. You have a sensor in your medulla oblongata that is being triggered by those baroreceptors. And it says you need to initiate a sympathetic fight or flight response because blood pressure is falling. And your body is actually very keen on keeping your blood pressure constant. So it triggers a sympathetic surge, and have you ever noticed that your heart may be racing for no reason, maybe, you know, in the middle of winter if you're very dehydrated? You may have some tachycardia as a result. I have a lot of people that ask me that too. Why do I have such a high heart rate? And I usually say, you probably need to drink more water, right? Because you have very low blood pressure. Okay, so what is your body trying to do? That's kind of teleological thinking. Your body isn't trying to do anything, right? These are just mechanisms in place that are triggered. But your body is sending a signal to the cardiovascular control center to increase heart rate, increase stroke volume, cardiac output, to get your mean arterial pressure back up to a homeostatic range. Okay, opposite is true if you have a surge of high blood pressure. It's going to send a signal to the cardiovascular control center to increase the parasympathetic response so that you lower heart rate, lower cardiac output, so you bring that mean arterial pressure back down to normal. Physiology is really just all about a way of thinking, right? So it's understanding these control systems and having an understanding of homeostatic ranges and mechanisms that, right? That's what physiology is about. Yes?
So if the mean arterial pressure is low, would your body have an increase in sympathetic or just a decrease in parasympathetic?
Both. That's a really good question. Usually it increases sympathetic, but it's kind of like the gas and the brakes. Increase the gas pedal, take your foot off the brakes at the same time. Yep, really good question. And that actually amplifies the response a little bit too. So you can actually see here, now this makes a little bit more sense right here. So when you have hemorrhage, all these different parameters play a little bit of a role in trying to get your mean arterial pressure back up to normal. Stroke volume plays a little bit of a role. These are reflex compensations. Heart rate, a little bit. Cardiac output, even more. You get a little bit more vasoconstriction, so the total peripheral resistance goes up. And all of these together, you can see, have a profound effect on mean arterial pressure. Okay? So it's, again, trying to get that mean arterial pressure back up to normal ranges. And all of those play a role. All of these mechanisms come together to try to get your mean arterial pressure back up. All right, right at 5 o'clock, you guys. I'm impressed. Good job. I will see you tomorrow. Hopefully this was helpful. I'll put the top hat questions up. And I'll